Students, today we will perform DLC, Differential Leukocyte Count. This is your Leishman stain, which is made up of eosin, methylene blue, acetone-free methyl alcohol. This is distilled water. This is staining rack. This is glass slide. This is your pricker and cotton. Let's start the practical. First we'll sterilize the finger and then pricking. Don't squeeze your finger. Discard the first drop of blood. Now lift a slide with your other hand, holding it along its long edges, and touches the top of the blood on the down surface about 1 centimeter from its narrow end. The drop to the right side. Hold the left end of the slide with the thumb and the finger of your left hand. Now grab the long edges of spreader lie between the thumb and the finger of your right hand. Now place the narrow edges of the spreader on the first slide in the front of first drop, making angle 45. Move the spreader backward so that it touches the blood drop. Hold it there till the blood reaches at the end of the spreader, and gently spread forward to the left in a single smooth motion like this, and maintain 40-degree angle. Drying the film by waving the slide. The ideal blood smear should occupy the middle two-thirds of the slide. It should be tongue-shaped, no striation, neither very thick nor very thin. It is buff-colored. Under microscope, red cells are separated from each other and no role formation. आप ये स्लाइड्स रख देनी है जब तक completely dry नहीं हो जाती. Our slides are completely dry. Put the drop of the Leishman stain to cover the entire surface, and stain should be added from the side to cover the smear. And wait for 2 minutes. Add distilled water, double to the Leishman stain. पहले 6 drops डाले थे, अब हम 12 डालेंगे. No, 11, 12. And wait for 8 to 10 minutes. It's depend upon temperature. अगर winters हो तो winters में हम 10 से 12 मिनट लेते हैं. अगर summers हो तो उसमें 8 to 10 minutes. With the help of pipette with both sided open, and blow the slide to mix the smear and distilled water like this. and wait for 8 to 10 minutes. 10 minutes is completed. Then we watch the slide like this at slanting position. Slanting position में इसको slide watch करनी है. Back the rub of the slide. The slide should be watched at least 2 minutes. और हम इसको ड्राई करने के लिए रख देंगे. Then after that we will observe under the microscope. Under high magnification, the red cells are stained orange pink and appear as numerous non-nucleated and bioconcave discs. Other leukocytes. There are mainly 5 types. These are neutrophils, eosinophils, basophils, monocytes and lymphocytes. They are all larger than red cells. These are depend upon size, nucleus, cytoplasm and cytoplasmic granules. Draw a table of 100 squares for recording various WBCs. Now start from the upper corner of the smear and move slowly to the right. Identify and enter the WBCs as they encountered using the letter on the counting the cell in a line until to reach the other end of smear. Now 2 fields down and scan the film in opposite film, continuing the identification of WBCs and entry into your table. As we reach the head end, again move 2 fields down and scan the smear towards the tail end in this method till you have examined 100 cells. When counting has been done, calculate the percentage of each type of cell in your count of 100 white cells. Draw a table of 100 squares for various WBCs. Identify and enter the WBCs using the letter N for neutrophils, M for monocytes, L for lymphocytes. Lymphocytes में हम सिर्फ L का लेटर यूज करेंगे, स्मॉल हो चाहे लार्ज हो, हमने सिर्फ L डिनोट से करना. तो हमारा पहला सेल आ गया न्यूट्रोफिल, देन अगेन न्यूट्रोफिल, नेक्स्ट lymphocyte, again neutrophil, lymphocyte. Our table is completed. N for neutrophil 55%, eosinophil 2%, monocyte 5%, lymphocyte 38%, and basophil not seen.